degrees, yes. So it's really nice. Well, I will start with the presentation, just not to delay things and honor the people that is connected in time. So it is, of course, my great pleasure and an honor to welcome Dr. Eric Brown to this 2020 Argentinian Society for General Microbiology series of conferences. We are, as I told you before, enormously grateful to you, Eric, for accepting this invitation. And before Dr. Brown's presentation, I would like to thank the SAMIGE members that make this organization possible, and to all the attendees for being here for this lecture today. So now let me now briefly introduce Dr. Eric Brown to you. Eric is a distinguished university professor at McMaster University at Ontario, Canada. He works in the Department of Biochemistry and Biochemical Sciences, and he is also a member of the DeGroote Institute of Infectious Disease Research. Dr. Brown is a fellow of the American Academy of Microbiology, and he has received numerous awards, including the Canadian Society of Microbiologists for Career Achievement, and from the Canadian Society for Molecular Biosciences. He currently holds a Killiam Research Fellowship from the Canada Council for the Arts and a Canada Research Chair in Microbial Chemical Biology. He has published more than 180 scientific publications in the most prestigious journals, I can tell, such as Nature, Nature Microbiology, Cell, PNAS, and so on. And he currently is also part of the editorial board of many of these outstanding scientific journals. He has also been member of advisory boards for numerous companies and national and international associations, including the Institute of Infection and Immunity of the Canadian Institutes of Health Research. And now, regarding his specific area of research, the Brown's Lab main goal is to contribute to novel directions in antibacterial therapies. In this context, they develop creative new platforms in the area of chemical genomics, and they also work in the discovery for bacterial cell envelope and nutrient stress physiology. In this last aspect, the Brown Lab aim is to understand the potential of nutrient biosynthesis, you can correct me, Eric, please, as a new target in drug-resistant pathogens. And this will be the main topic of this talk today, as I believe. First, let me remind all the audience to shut their microphones then. And by the end of the conference, Dr. Brown will eagerly take your questions in the order that they appear in the chat. So please write your question or the word question in the chat, and we will tell you to open your mic if you want, or just read the question in the order that they appear. And well, the audience is all yours, Eric, and we thank you again for this magnificent opportunity to hear to your research.
Well, thank you. I really appreciate it. It was a very kind introduction and a kind invitation. I'm like everyone, I think we're all struggling with this pandemic, and it's nice to do some things that feel slightly normal, which is giving talks at meetings and gatherings such as it is. Yeah, we'll just have to make the best of it. Can you see my screen okay? Does that look good?
Yes, it's perfect.
Okay, wonderful. And you can see the cursor if I want to point a little bit with the cursor on my computer.
Yeah, we see the pointer also. Yes.
Perfect, okay.
Perfect.
So yeah, as you suggested, I'm gonna speak today about our work on in the area of nutrient stress, and come up with the cheeky title, Starving Bacteria to Find New Antibiotics. I'm I'm working at my standing desk today because it just doesn't feel right to give a talk sitting down. So I'm actually standing up so I can, if I pace out of vision of the camera, you can shout at me and just remind me to come back. Okay, slide advance, slightly different. So I begin with a slide which is a rather old issue of Time magazine. I'm a postdoc at Harvard Medical School in Boston, and I'm working in Chris Walsh's laboratory at the time, studying cell wall biosynthesis in bacteria. And quite frankly, I'm a young man. This is 1994, I believe, in the fall, and I really have no idea that I'm doing anything that has any practical utility whatsoever. And this appears on the cover of Time magazine. And we're very used to seeing these headlines these days. I mean, I dare say these things are coming, by the week quite often. And it was nevertheless to me, quite a surprise to see this on the cover of Time. Again, this is many, many years ago now. We have problems in the clinic at this point with organisms like methicillin-resistant Staph aureus, vancomycin-resistant enterococci, but they're really troublesome gram-negative microbes, right? Where we're learning there's the greatest medical need today in terms of new antibiotics. Bugs like Pseudomonas aeruginosa, Acinetobacter baumannii. These organisms were not, known to be trouble at that time, and so it's not hard to conclude, for example, that things are much, much worse now than they were in 1994, and there have been repeated calls for action from not just the popular press, but governments around the world, non-governmental organizations, including, of course, the World Health Organization, to try to call attention to this problem. And at the heart, of course, of this problem is the issue of antibiotic drug discovery. Now, there have been quite a number of reasons why antibiotic resistance has become a problem. Not the least of which is overprescription, the use in agriculture, and a variety of concerns that aren't really about discovery, and they aren't really about things that scientists think too hard about necessarily. But it really is illustrative if you look at the number of new chemical classes over the history of antibiotic drug discovery, parsed out here according to decade. You see that the preponderance of discovery is happening here in the 40s, 50s, 60s, and 70s. These are the new chemical classes, right, that wind up in the clinic as new drugs. This is not the date that they appear in the clinic, but the date that they're discovered. For example, vancomycin, daptomycin is discovered in the 1980s, or it appears in the clinic in the 2000s. But I'm growing up in this period, right, as a biochemist and as a microbiologist in this kind of modern era of drug discovery. And I mean, it's something, I see this as something of an embarrassment that there's been no success during this period. Exciting advances in recombinant DNA technology, in high throughput screening, high throughput synthesis, structural biology, and of course, genomics is huge during this period in terms of enabling genes to drugs and the efforts that have ensued in modern drug discovery. And yet there's been little or no success during this period, even in the way of kind of exciting things that might even be in the clinic right now. There's a real paucity of new chemical classes of compounds to treat drug-resistant infections. And so this is the group. We're about 20 these days. This is taken about a year ago at a lab retreat that I run every year. That sadly we were not able to have this year. We're planning a Zoom session of our own to substitute as a retreat this year. But I work with a fantastic group of young people. We have pictured here postdocs, graduate students, some undergraduates who do their thesis work in my group, and some really valuable staff members and research associates. They're working in three areas: nutrient stress that I'll talk about today, also cell envelope system. We have a pretty significant effort trying to figure out ways to disable the outer membrane of gram-negative bacteria to enable the passage of drugs better into gram-negatives. And increasingly, we're using tools of systems biology in my group to try to kind of get away from genes to drugs, reductionist drug discovery, toward a more holistic approach to understanding the biology and toward discovering new and interesting targets and chemical matter. So you know, most of the success in drug discovery has happened during this era where much of the initial phases of drug discovery have really been done by screening, quite frankly, soil samples for the ability of soil-dwelling bacteria to produce compounds that will work on a petri dish. And so we have this marvelous paradigm that emerges, you know, that really underlines kind of an approach that all drug discoverers now use, where we have these valuable in vitro assays that then are tested in animal models of therapy, and then ultimately translated into humans. And quite frankly, the antibiotic drug discovery area is of really the envy of just about every other therapeutic area for the fidelity, really, of these steps. You know, for example, cancer researchers have cured cancer over and over again in mouse models, but those models really often fail to recapitulate what goes on in the human. I'm going to tell you today, as much as this model has been tremendously useful, it also has some very serious shortcomings. And in the first part of the talk, I'll talk about a story that really questions the validity of the petri dish as a good model for in vivo. And later in the second part of the talk, I'll tell you about our efforts really try to understand how we might do better than a conventional mouse model of infection to predict what might be the best compounds and targets that would work in a human. So we begin with Mueller-Hinton broth. This, of course, is the clinical standard for microbiology. It's the way that clinical microbiologists do their testing of minimal inhibitory concentrations to make decisions about what to use in the clinic. It is also what antibiotic drug discoverers use, you know, as a first pass to decide what compounds might ultimately have the most utility for the animal model and ultimately in the human. And so remarkable that this media was discovered in 1941, Howard Mueller and Jane Hinton. Jane Hinton, by the way, was an African-American woman who would go on to be the first Black veterinarian woman in the United States, graduating from the University of Pennsylvania, who was working in a lab at Harvard Medical School. It was kind of down the hall from her father's. But if you look at this media, you know, it was designed to grow fastidious organisms, meningococcus and the like, that wouldn't grow very well in any other media. 2 grams of beef extract, casein hydrolysate, of course, is a source of amino acids, starch in a liter of water, cation adjusted, has magnesium and calcium. And this media has become the standard, right? And certainly in the United States and Canada, where, you know, we think about the Clinical Laboratory Standards Institute as sort of the organization that tells us how to run these things. All of this is run around MHB media and MIC testing. And so, you know, I'll foreshadow here that, you know, we're asking a lot in my group whether or not this rich microbiological media or anything like it for that matter is really a great proxy, you know, for what's befallen this poor individual in terms of trying to decide what's, you know, what are the right targets, what are the right chemicals. So return again to this timeline of antibiotic drug discovery. And, you know, when we started thinking about this problem, thinking that maybe rich microbiological media wasn't wasn't necessarily the right way to go. You know, it's instructive to look at the way that antibiotic drug discovery has been done over the years. And of course, this is now the individual discoveries on that same timeline. Shown in the green here are the natural product compounds that were discovered from soil-dwelling microbes. And those in a lighter yellow are the synthetic compounds that, that have been discovered over the years. All of these, except for one, were discovered using rich microbiological media. And if we were all in the same room, I'd ask someone to throw up their hand and tell me which one. But we've got sulfonamide really being the only molecule that wasn't discovered in this way. And it really is remarkable to think that this rich microbiological media has really become the standard. There's another great statistic, and that is that if you allow E. coli to grow in exponential phase with a doubling time of 20 minutes, it would only take 2 days for one organism to equal the mass of the Earth. And so clearly that's not what's going on. Maybe we can do better than that. We take some inspiration from work from this man, Gerhard Domack, who was working at Bayer in Germany, and in 1932 discovered this compound, Prontosil. It's a sulfanilamide version of the sulfonamide molecule. This molecule came from a screen that Domack was doing of dyes that at that time Bayer was part of an amalgam of companies that included several companies that made dyes for the textile industry. And so Domack's job was to screen those dyes looking for therapeutic value in some of those. He found that this molecule could stain cells red, but did nothing on a dish that contained rich microbiological media. And then he had the audacity to systematically start testing these compounds in an animal model of infection, despite the fact that they didn't work on a dish. And that's how he discovered the activity of Prontosil, which is in fact a prodrug that is yet another level of complexity here, where there needed to be some physiology to act on this bond in order to produce the active compound. That earned him the Nobel Prize in '39. The German government allowed him to accept that prize in '47. And you know what's amazing is that there's been no one has learned this lesson, that in fact a molecule that doesn't work on a dish could be quite effective as an antibacterial really in the 80 years since. That is, we have not seen any other antibacterials discovered in this manner. Here's a look at what sulfamethoxazole, kind of the modern day antifolate in the pathway at axon, begins with charismate and GTP, and then we have the bond formation between these two compounds, pyramidal and this hydroxymethyl dihydropterin diphosphate compound to form 7,8 dihydropteroate. This carboxylate that ultimately is important for the addition of glutamate and key for the creation of tetrahydrofolates that are so important in, for example, thymidine synthesis and the synthesis of methionine, for example. But this molecule is a kind of a Trojan horse molecule that looks a lot like pyramidal but is incorporated by full P into an intermediate that can no longer go ahead to make tetrahydrofolate. It's a really successful and interesting example of a compound that's still used in the clinic today, in combination, of course, with trimethoprim. So we're thinking about this problem that maybe we don't have the right target set necessarily. Our favorite model is E. coli, and of course, E. coli is a gram-negative bacteria that has 4,400 genes. And Hirotada Mori, working at the NAR Institute some years ago, showed that 303 genes resisted deletion and were essential for growth on a rich microbiological media. And if one asks that question instead on minimal microbiological media, where you would just have, say, glucose, ammonium chloride, and trace metals provided to the bug, it's 422 genes that resist mutation. So that's 119 genes that are specific to an essential role on minimal microbiological media. And they are in the areas of physiology that you might predict, particularly amino acid biosynthesis, some 57 genes, 21 genes involved in nucleobase synthesis, and 19 genes in vitamin synthesis. And then some others that are curious, some of them explainable, some of them still a little bit enigmatic to me. But we have decided in my group to really focus on these three pieces of the puzzle here: amino acid synthesis, nucleobase, and vitamin synthesis. And to Sumayya Zlitny, who is a PhD student in my group, who did really some of the first work to come up with a workflow, a platform that we could use to screen for compounds that would be active against bacteria growing on minimal microbiological media. So the first screen she did was kind of a pilot screen on a diverse library of 30,000 synthetic compounds growing E. coli and glucose and ammonium chloride. And what she found was that, and it's an assay that we did in triplicate, in fact, so it's shown here, it's sort of 3D. The blue compounds are those that don't have any impact. The red are those that show some growth inhibition. She found some 340 molecules were novel and had activity against E. coli under these conditions. She was interested, of course, not in the ones that would inhibit growth in rich media, but those that were restricted to minimal media. And so what she did was she added back nucleobases, vitamins, and amino acids, and prioritized 71 of those that she was unable to block the growth of. And those then could be subject to follow-up using a platform that I'll show you. But here's a look at a keeper molecule where we're looking at the growth of E. coli as a subject of compound concentration in minimal media. This compound 6191 that has an EC50, right, for its inhibition of growth around 0.5 micromolar. And then when we add back nucleobases, vitamins, and amino acids, you see this activity is completely suppressed. And so this is a molecule that 6191 was one that Sumayya kept and moved along in the process. The next thing we realized quite quickly is that we could prioritize molecules that could be suppressed by amino acids, nucleobases, and vitamins. And that allowed us to try to generate some hypotheses about which of those pathways were targeted by a particular molecule. And so she came up with what we call the metabolite suppression array, which is about 1/2 of a 96-well plate, where we had a well, of course, that has no supplements in it. That's the M9 well, where we would see growth inhibition routinely, adding back all nutrients, adding back just amino acids, just vitamins, just nucleobases, combinations thereof, individual amino acids, nucleobases, and vitamins, et cetera. And so this was the idea here is we could generate some hypotheses about what the targets were for these molecules. She validated this idea using known probes. So here's a heat map in which dark blue is no growth in the presence of a compound, and bright yellow is 100% growth with various sort of degrees in between. And what we have here on the on the columns are the various media. M9, where we have, of course, no growth throughout, adding back all nutrients, just amino acids, just vitamins, just nucleobases, et cetera, amino acids, nucleobases, vitamins, and PABA. You can see that conventional antibiotics like tetracycline, chloramphenicol, and kanamycin, of course, you can't suppress the activity of these molecules by adding back nutrients. And so the idea to kind of test here was, does this work with known probes? Molecules, for example, that would inhibit aromatic amino acid synthesis, folate synthesis, pyrimidine and purine nucleotide synthesis, et cetera. And if we just key maybe on our favorite here, sulfamethoxazole, you can see that, of course, there's growth inhibition in M9. It goes away when you add all supplements. Adding nucleobases doesn't do anything here, but pools otherwise thereof. Methionine and PABA are key signature suppressors, right, for the action of this compound. Methionine, of course, getting its methyl group from the methylated folate. So here's a much larger heat map now, where we're looking at all 71 of these compounds along with known antibiotics. We have here, of course, a signature in each case of the action of each of these compounds on E. coli, where we have signature suppression signatures when adding back these various nutrients. And I'll just talk about three sort of areas on this plot to start. The first is that of sulfamethoxazole, where we can suppress, of course, with all nutrients. There's methionine, PABA over here. And what we saw is that a bunch of these molecules, a remarkable number of them, registered and clustered along with sulfamethoxazole. And it turned out, and we didn't know this at the time, but that among the 30,000 compounds that we screened, there were a whole bunch of para-amino sulfonamide containing molecules. Some, what do we have some 15 compounds that shared this structure. And so that was news to us. I mean, it suggests to us that one of our compound suppliers had been, working on antifolates. But we also had this particular compound over here that was quite novel in its structure. Turns out that molecule, in fact, is an inhibitor of PABA synthesis. It inhibits that first step in going from charismate to the dihydropteroid intermediate. Reasonable Ki, it turns out to be a slow binding and reversible inhibitor with a reasonable inhibition constant for the growth of E. coli and some significant activity against Pseudomonas aeruginosa. Our work kind of continues on this compound, but I won't tell you much more about it, except to say perhaps that medicinal chemists don't love this compound. It has some sort of offending chemical features like a nitrobenzene group and a group here which has some reactivity. But I'll point out that many of the compounds that we pull out of our screens seem to be compounds that will covalently inactivate their targets. And we think that might be because the cells are really telling us what's going to get the job done here. And it might be that things that bind in an irreversible fashion are better inhibitors, and they can't be competed out when substrates higher in the pathway would accumulate and might force off and compete with an inhibitor that is otherwise not covalent or irreversible. There's another interesting category of compounds here on the heat map that clusters with glyphosate. Glyphosate is better known in some circles as Roundup, that inhibits aromatic amino acid biosynthesis. And we've reasoned that we might have in this instance compounds that are hitting high in biosynthetic pathways where they would wind up with multiple end products. And so that it's important to think about how to better suppress the action of those. And we came up with metabolite suppression array version 2. That has not just individual amino acids, vitamins, nucleobases, but in fact has pools thereof and a kind of a signature collection of intermediates. This particular strategy, which is now a 96-well plate, came in fact after Sumaya disappeared to the library for about a month studying the oxytrophy literature that's probably 40 or 50 years old right now. But it works well to this day, of course. So one more that I want to key on here before I kind of move on to the second part of the talk, and that is this compound 13772 that was absolutely unique in that it was suppressed by vitamin B7. So we just show that here sort of in a blow up. Again, this compound is effective in M9 and is suppressed with the addition of all nutrients. Amino acids are not doing it. Vitamins are suppressors, nucleobases are not. And the vitamin that suppresses here is vitamin B7, better known as biotin. There's the compound, and it has this hydrazine moiety, also not a med chemist dream compound, but hydrazine moiety out here and a nitrobenzene group, nice and compact, remarkable activity on E. coli and some interesting activities you'll see on other gram negatives. So that was exciting to be able to really sort of generate quickly a hypothesis about a compound and come to the conclusion that biotin is very likely the target. But Sumaya got even more lucky than that in some respects. There's the pathway for biotin synthesis, begins with familial ACP, generates kappa through the action of BioF and the addition of this piece from alanine to give a diamino compound, diaminopelargonic acid. The top end of that ring is built with the addition of a CO group from CO2 to form dithiobiotin, and then biotin around it with the addition of the sulfur from S-adenosylmethionine by BioB. It turns out that that deletions in each of these genes, BioF, BioA, BioD, and BioB, can be, the oxytropies that are generated on minimal media can be solved with the addition of the downstream compound. So a deletion in BioF can be, the growth inhibition that's generated there in minimal media can be solved with the addition of kappa, likewise DAPA for BioA, DCB for BioD, and biotin for BioB. So that enabled SAMIGE to have a careful look at the suppression of this compound, 8 microgram per mil MIC on minimal media in the absence of supplements, greater than 256 micrograms per mil. That's more than a 32-fold suppression by the addition of supplements. The addition of kappa did not change the MIC, so a fold suppression of 1. The addition of DAPA, DTB, and biotin, however, each led to a 32-fold suppression. And so, of course, what this suggested was that, in fact, BioB, or BioA, pardon me, was the putative target of this particular molecule. And sure enough, SAMIGE made recombinant BioA protein, developed an assay for that transformation. And if we look at the compound concentration against activity of that enzyme, it has a midpoint of approximately 250 nanomolar. So a relatively potent starting point for a compound that is on target for an enzyme and vitamin synthesis, and is of course working against a whole organism. We were interested in kind of how this compound was exerting its inhibitory effect, and we had a hunch that this hydrazine moiety may have some reactivity, a kind of a soft nucleophile, if you will. Obviously not wildly reactive, because this compound is specifically suppressed by biotin, so it's not kind of willy-nilly reacting with enzymes in the bug. But what we did find is when we capped this hydrazine with an acetyl group, the activity completely went away against the bug and the enzyme. And likewise, if we substituted this hydrazine with a hydroxyl moiety, the activity also went away. And so we knew well that this enzyme, BioA, was a pyridoxal phosphate containing enzyme, right? Where what happens is you park an amino group at this site that then is picked up by to form the diamino compound. And so we had this idea that kind of probably what's going on here is we have attack by the hydrazine group on this internal aldimine, where the lysine of 274 of BioA is holding the pyridoxamine group at the active site to form this adduct that could well then be sort of irreversibly bound or tightly bound by the enzyme. And sure enough, when we looked at the adsorbance of enzyme on addition of MAC13772, we see a blue shift. So the without 13772 is this red trace, and we see a blue shift and an increase in absorbance that coincides precisely with one equivalent of 13772 per equivalent of BioA. So seeing is believing, and at the time we actually an undergraduate who was working in my laboratory went down the hall to Murray Junip's laboratory at McMaster and came back to the Brown lab with this particular structure. So it's a beautiful structure of a mostly alpha helical protein, 420 amino acids per subunit, bound as a dimer, where the active sites are actually in 2 locations at the subunit interfaces. And so some of the residues in the active site are highlighted here. And if we do a close-up of that active site, we see that there indeed is the 13772 molecule bound in covalent linkage with the pyridoxal phosphate cofactor, where this lysine 274 that was otherwise linked to PLP is now released. And you have this thing bound in a relatively hydrophobic pocket surrounded by aromatic amino acid residues. And we've been hard at work at this since, really trying to imagine how we might build a better and more potent 13772. We are we're also encouraged by the idea that natural products are also inhibitors of biotin synthesis. So antibiotic drug discoverers like to think that natural products validate targets. And in fact, often if, you know, we're a research group that likes to think a lot about novel targets, and often I'll be talking about a target of utmost novelty, and frequently I'm challenged, well, there's no natural product inhibitor of that enzyme though. Are you sure it's a good target? Because maybe, you know, if nature hasn't chosen it, maybe it's not a good one. Sure enough, BioA is a target for nature. And you have these two molecules, this N-methylvaline and N-methyl isoleucine compound that are linked to this unnatural amino acid with this cyclohexadiene group that activates an amine. It turns out that the amine in these amyclonomycins works exactly the same as the hydrazine that I showed you. So it's a soft nucleophilic nitrogen that through aromatization of this ring, you know, wants to participate in a bond and does so with PLP. And so here are a couple of structures. Now we're comparing our 13772 addict with PLP with another structure that we've generated in my group, not published yet, of the serodentium BioA, which is tightly bound now to this N-methylvaline version of the amyclonomycin analog. So we're pretty excited about these directions, that we have these two probes, which are, you know, useful starting points perhaps for new drugs, new leads that would target BioA. But I want to tell you about work that we've been doing more recently that has really been led by Lindsay Carefree, a PhD candidate in my group that was just published this year. And the question really that Lindsay's been trying to address is, we have these great molecules. Are they really useful as starting points for new antibiotics? Is biotin really a good target for antibiotic drug discovery? And in fact, there was some suggestion that, you know, biotin might not be great. We're now kind of examining this step, right? You know, what's kind of going on in the mouse. There have been several studies, of course, where folks have created large mutant collections of various pathogens. You know, what's often done is those collections are introduced into mouse models of infection, and then we ask, you know, what mutants can we collect from the mouse? And if genes, for example, in biotin synthesis are missing from those that we take out of the mouse, we come to the conclusion that those particular mutants were unable to survive in the mouse, either for colonization or persistence. And it turns out, if you look really into the fine print of those kinds of papers, you don't often see biotin genes coming up as important for colonization and persistence of the mouse. And so I'm foreshadowing here a little bit is that, you know, what I'm going to tell you is that Lindsay's really gone deep on this to suggest that maybe those experiments are not quite perfect, and that this mouse model, this conventional mouse model of infection, may have some shortcomings, particularly in the area of nutrient synthesis. So one of the first experiments that she did was this one. She asked, really, you know, very simply, if we examine the growth of a BioA deficient E. coli, how much biotin does it take to support the growth of that mutant? And it turns out there's an inflection point somewhere around 0.1, maybe 0.2 nanograms per mil of biotin. And it's quite a, you know, a clear inflection point. We can very clearly define what the biotin needs are of a BioA deficient E. coli that can't make biotin. She looked at a bunch of other bacteria, Klebsiella, Salmonella, uropathogenic E. coli, A. baumannii, Pseudomonas aeruginosa. And interesting that the baumannii and the Pseudomonas are kind of separated from the others. They're out here, right? Where maybe it's because these bugs are really environmental microbes that they have higher, you know, requirements perhaps for biotin, and really rely on synthesis as opposed to sort of, you know, grabbing it from the host. But if we look now, pardon me, if we look at the concentration of biotin that we typically see in the mouse, it's a band that kind of spans somewhere between about 5 and 20 nanograms per mil of biotin. And so clearly that's an amount of biotin which is really going to rescue any of these auxotrophs for biotin. But The human, as it turns out, has a band of biotin which varies somewhere around, 0.1 nanograms per ml up to about 2, which suggested that, at least some of these organisms may have biotin requirements which exceed that of what is typically characterized for humans. And so Lindsay, wanted to look a little bit closer to this and get more still in an in vitro system, but she decided to look in human blood. So what was the biotin synthesis that typically supported the growth in serum from human blood? And so we're looking at an experiment now where she inoculates a tube containing wild type bacteria or biotin deficient bacteria into human serum, and then asks 24 hours later, what was the growth? And of course, if it's an A. baumannii, after 24 hours of growth, we see, something like more than 1,000 times as many bacteria over that period. But in the absence of BioA, the growth is completely halted in human serum. Klebsiella pneumonia, a little bit more growth, but well short of what we see for wild type. Same situation for Pseudomonas and S. typhimurium, suggesting that in all of these bacteria, in fact, there's insufficient biotin in serum to support at least wild type levels of growth. And of course, when she looked at mouse serum, it was a completely different story. Whether or not it was a wild type bacterium or a BioA deficient bacterium, these grew equally well in serum that was collected from mouse as compared to humans. So suggesting that there's kind of a wildly different situation going on in human and mouse, of course, where mouse is where most of the target validation has been done to date on virtually all genes. So she got to thinking and knew well about this phenomenon. So this is the growth of an E. coli biotin mutant on nutrient-rich media. And if you add streptavidin to a nutrient-rich plate, that will suppress the growth of this strain. And that's, of course, because streptavidin binds very, very tightly the biotin that's in that media and doesn't allow E. coli to use it. In fact, it's thought that streptavidin is a kind of antibiotic that's produced by Streptomyces bacteria that's often produced actually in the same strains that are making these amikalinomycin molecules. So kind of a double whammy coming from the producer strain. And so Lindsay had this idea. It was, could we inject a mouse with streptavidin and make it more human-like so that we would have now a reasonable model in the mouse that would mimic the situation that's in the human. So here again is that band of biotin that we know is typical of the human, somewhere between sort of 0.06 nanograms per mil to 2 nanograms per mil. There's the level of biotin that's in the mouse without any streptavidin. And what Lindsay was able to do was add 2 mg per kg of streptavidin in this mouse model and record levels of biotin over a total of 12 hour spread, which was she was shooting for a time regimen that would be useful in the context of animal experiments that she would do down the line. And what she found was for at least 12 hours, she was able to maintain biotin levels that were relatively similar to that seen in humans. And of course, this allowed her to do a mouse mimic of human infection. And so the following experiment was one of the first that she did after that success that I showed you on the previous slide. She did a competition experiment where you take a strain of E. coli that is deficient in BioA and you co-infect with the wild type strain, baumannii, Klebsiella pneumoniae, Pseudomonas aeruginosa, and then ask after sacrifice, what's the ratio of those 2 strains? BioA deficient divided by wild type was close to 1 for all of these particular bugs with the conventional mouse model of infection. And of course, if she's now adding her streptavidin to make a mimic of human infection, there's just a huge disadvantage for the BioA mutant, for the A. baumannii strain, about 1.5 log for the Klebsiella pneumonia strain, and about 1.5 log for Pseudomonas aeruginosa. So these are pretty exciting findings to us. We had, of course, at our disposal this compound MAC 13772, and we were very interested to know, could this compound be efficacious? And I'll tell you that, we discovered this compound some years ago and had been collaborating with folks at the NIH to try to investigate things like pharmacokinetics, efficacy. We'd done mouse model of infections. They'd failed for us. And we also did quite a bit of PK work on this compound. And what we found is that its PK wasn't very good. That is that it was cleared very, very quickly and didn't really reach levels in the mouse that would lead us to believe there would be efficacious quantities. But what we didn't know, of course, at that time is about the high levels of biotin that are in a mouse. So Lindsay was keen to revisit this. Abelmanii infection of the mouse, MAC 13772 is a therapy. Again, there's the standard kind of conventional model, vehicle 13772, not a lot of difference in terms of the CFUs per mil for blood. And here's what she saw when she made the sort of humic mimicking levels of biotin in the mouse, that indeed there was about a log and a half effect, which we were pretty happy with, because we know that this molecule is, really has pretty terrible pharmacokinetics. So we're kind of excited to move ahead with this molecule and think about how we might make it better, how we might make a better molecule, for example, from a pharmacology standpoint, and work is ongoing there. We're also screening lots more synthetic compounds. We have a library at McMaster of nearly 500,000 compounds, diverse synthetic compounds, and we're moving through that entire chemical collection now, using this assay, looking for growth inhibition on glucose in the presence of just, ammonium chloride as a source of nitrogen, trace metals. We've generated some 9,000 actives in those screens and prioritized about 1,000 compounds to use our hypothesis generation tools. And we're systematically working through these compounds to find more probes of nutrient stress that who knows, may also have value as leads for new antibiotics. And so I kind of want to finish with this idea about druggable genes and innovation risk. We work a lot in my group. We've had lots of good collaborations and interactions with the pharmaceutical sector over the years and biotech companies. And there's a very interesting conversation that goes on around, what really is the druggable genome of a bacterium. And of course, academics like myself like to think about, what's important for growth in the human. That's the in vivo set of drug targets. That's this target set number 2. And of course, drug discoverers have really emphasized over years this set of genes. I told you at the outset, at the top was about 303 in E. coli, right, that are important for growth on rich microbiological media. And of course, this set is really a subset of those that are required for the growth in vivo. We don't know really how big this circle is, but we know it's much, much bigger than this circle from a lot of the transposon mutagenesis experiments that I talked about earlier. And of course, drug discoverers will remind us repeatedly that there is tremendous innovation risk in targeting these in vivo genes. Many of them we can't build in vitro assays for, so we can't really do the biochemistry. We can't really do the work on a dish in vitro in preparation for the animal model. They're just about as far as you can get from that MHB plate and the Clinical Laboratory Standards Institute obsession with growth on MHB and the ability to characterize an MIC in vitro. And so there's great reluctance really to try to move on this target set. And so we've been thinking, more recently that this group in nutrient stress, maybe it's sort of intermediate risk. It's not a kind of a no-brainer collection of targets such as you have for one, but there are some real advantages to this target set number 3. I mean, many of them are of course genes that encode enzymes that are involved in the synthesis of metabolites that are well understood. We can build biochemical assays and we can do some great in vitro biochemistry around them. They're typically small soluble proteins that are amenable to the sort of structural biology that I showed you earlier. But also if we build nutrient specific media, right, we can look in vitro at the growth on a dish and do those characterizations. They might not be CLSI, that might not be MHB, but there's an opportunity at the very least for the characterization of that particular target and its importance in bacteria in vitro. and maybe even as we have seen with the biotin, an opportunity to build specific, perhaps non-conventional, animal models of infection. We need to accept, I think, though, that of this set, again, which would include this and somewhat be part of the in vivo set, that there's probably a bunch of them that are really not practical in vivo. That is, they're dispensable for growth in vivo, but that genes like biotin synthesis, are clearly in this territory. So just to summarize, then, we really think that nutrient-limited growth conditions are fertile ground for the discovery of new growth inhibitors, and that this platform we've come up is really a great way of providing strong mechanistic hypotheses to go forward with. We have a couple of probes, maybe even leads for new drugs in the area of biotin and PABA synthesis, and we're now going into large-scale screens for both synthetic and natural product chemicals at this point. Biotin synthesis looks like a good target for new antibiotics that's been underestimated by conventional mouse models, and that in all these new compounds, if they're anything, I think they're good tools for target validation, and may even have, in some cases, potential as leads for new drugs. I showed you the picture of the group earlier. We're grateful for funding from a lot of Canadian funding agencies. I think this is a list more or less of virtually every company, biotech and pharmaceutical company that I've worked with over the years as either an acknowledgement or disclosure or both. And I think I mentioned most of the characters here that were important in leading the various studies, but they're shown also here. Structure Bioaid in a collaboration with Murray Junot, and phylogenetic analysis done with Caressa Tsai, who's part of Brian Coombs' lab, who I understand was with you maybe this time last year. So with that, I'll thank you again for the opportunity to be here, such as it is virtually, and take any questions.
Okay, thank you very
I'll stop
much.
Sharing, maybe, if that helps.
It's okay.
Yeah.
Thank you very much for a great talk. These discoveries are really nice, and the strategies that you used here, the ones that you showed here, and the one that I happened to read in your papers are really amazing and innovative. Actually, the way you look things is really different. So, well, we can take the questions if you want. So I will see the chat. Someone has questions for you. Diego, if you want to start with a question, you can unblock your mic.
I think Claudius first. Can it be?
Oh, great.
Just a question.
Oh, sorry. Yes.
Yeah. So I give the place to Claudio, and then.
I'm sorry, it was the other one that said, great talk, thanks. And I thought they were. Thank you. So,
yeah. So, hi, Eric. Yeah. Thank you for the talk. It was just great. I have written. It's one question. Have you challenged this compound that targets BioA in the context of a biofilm, an established biofilm?
Right. Yeah.
If so, how does it perform?
Yeah, I don't think, Claudia, that we've done that experiment, but I'm expecting that what we have shown is that biotin biosynthesis inhibitors are in fact static and not cidal. Lindsay's doing a lot more work these days where she's looking at combinations of biotin synthesis inhibition with other compounds. And actually, she's really working on a really exciting story that I won't give away the punchlines today, but things like resistance generation and other states of physiology have been great interest to her more recently. But I don't think we've done the experiment where you establish a biofilm, you treat with 13772. My expectation is that it wouldn't clear a biofilm. It's more of a static molecule better at growth inhibition.
Okay, thank you for the thought.
Okay, then Diego Serra.
Okay. Hi. It was a great talk. I enjoy a lot. It's really amazing work. Yeah, my question is a little bit related to the one from Claudio, and also because I also work in biofilm, but I mean, or related to stress responses when cells start to grow slow. So my question is, so in the case of biotin, you have to transport inside across the cell. So how this molecule, which are kind of analogous or intermediate in the pathway, enter into the cells? And I will imagine that growing slow, you have this compromise. This is one question, and the second one, if they allow me. So also when cells start to grow slow, at least in the case, for example, of E. coli, you start to protect your cell wall, and you have to produce extracellular metric, and so transport for something, it's much more difficult. So I will imagine some penetration problem that could be related to biofilm at the single cell, but also at community level,
Yeah, for the growth inhibitor, you mean?
Yeah.
Or do you mean for acquiring biotin from the environment?
Well, yeah, I mean, I will imagine that when you grow slow, at some point also you start to decrease the demand of biotin,
right, yeah.
All right? So
yeah, so we haven't, so these are great questions. The biofilm question, the sort of slow growing persister kind of, thinking. And we're interested in these phenomena, but I must say, I don't think we've done very much on the biotin inhibitor in this area. You raised a really interesting point earlier, though, about acquisition of biotin and biotin intermediates. We also did some work where we systematically knocked out. We did a cross of nutrient stress genes with the rest of the genome in E. coli. That's kind of in our systems biology effort, but we focused on nutrient synthesis because we were interested in what are the synthetic lethal interactions, say, of all nutrients biosynthetic genes. And, without fail, if you go through the list of genes like biotin synthesis genes, you come up with transporters as synthetic lethal interactions. So you can exist on rich microbiological media if you have biotin synthesis or if you have a transporter, but you can't exist if you don't have both. And so those interactions are often synthetic lethal. And we've actually used that interaction to grow cells on rich media to look for natural product inhibitors. One of the challenges in natural product extracts is they're full of nutrients, and our nutrient suppression methodology wasn't working very well. So we've used that synthetic lethal interaction between transport and biotin synthesis to allow us to, to discover the compounds that might be active against biotin synthesis. And I'm trying to remember whether or not in those synthetic lethal interactions, do they include some of the other genes that you're talking about? That is BioF on down to the ultimate biotin synthesis enzyme. Are they also interacting with those genes? Actually, you could have a look at that paper, in fact, and have a look at the data sets and make that answer yourself. The prediction, of course, would be that there'd be synthetic lethal interactions between those intermediate enzymes and the transporter, if it's the same enzyme. Or you may find a new transporter through a different synthetic lethal interaction.
Okay, very interesting. Okay, thanks very much.
Diego de Mendoza, you have the
yeah, thank you for your talk. I am wondering if your mutant bio-A is rescued by fatty acids, because I mean the only, I am wondering if fatty acid from the serum will not rescue the bacteria.
Yeah, it's a really great question, right? The single essential function of biotin is in fatty acid synthesis.
Oh, sure.
And in fact, this is something that Lindsay's looking into a lot these days, and she's doing these experiments she's finding, you know, phenotypically, there are very similar phenotypes characterized in different ways between the inhibition of biotin synthesis and the inhibition of fatty acid synthesis.
Yeah, sure.
So your question's a really good one. I think the oxytrophies around fatty acid synthesis are kind of tricky in E. coli. They're maybe you can maybe you can guide us on this.
It's very easy, I mean, to work with fatty acid also trophy in E. coli.
Yeah, so we should
according to my experience, of course.
We should look at this.
Yeah.
It's a great question.
Fernando
Yes, very do you hear me? Very nice question. Very nice talk. I have just a question. You show that in rich media you have one phenotype, and in minimal media you found this effect for this compound, but they were rescued somehow. If you grow the cells in serum, it depends also in which kind of serum. No, if you were using human serum, you have one phenotype. If you were using mice serum, you have another phenotype.
Yeah.
Have you tried to reanalyze the library in those serum, or in more, I can say, in more in vivo conditions?
Yeah, so we have, so another postdoc in my lab, Brent Weber, in fact, just published a paper, it's out in Cell Reports recently, where he did a screen in serum. This is kind of prompted our interest in this. This is, you know, a lot of the people in my lab are thinking now a lot about this, you know, how well the dish is really a mimic for what's going on, and what can we do in vitro. Serum is a really straightforward in vitro medium to work with. It's expensive, but it's, you know, it's doable. Brent took an ordered collection of transposon mutants from Klebsiella and tried to grow all of those in serum. And so he came up with a list of, you know, of all the genes that are important for growth in human serum and Klebsiella. And then he did a chemical screen, and he screened more than 100,000 compounds, looking for compounds that would, you know, selectively inhibit the growth of bacteria in human serum compared to just say LB or MHB. And it's an interesting paper. He's got you know, several little stories in there about compounds that have selectivity for growth in serum. It's an interesting, I'll make one more caution about that. And that is that one of the things he found, for example, is that a capsule mutant of Klebsiella will grow very badly in human serum. It'll grow just fine in mouse serum and doesn't grow well in rat serum. So, you know, it's the more that we look at this, you know, animal model situation, the more we I think need to be, you know, very, very critical and very, you know, careful about how to interpret these various results. And clearly mouse serum is also not a very good, you know, measure of the immune functions, right, that are present in the human. So there's, I think, lots of reasons to be careful about the mouse model.
Thank you.
Okay, if there's no other question from the attendees, I will be willing then to do my own questions here. And in the same line, I am curious about in which niches, apart from serum, you have tried these strategies. For instance, when bacteria grow intracellularly, which are your strategies towards for instance, an intravascular medium, or just to reach that point, and which are the auxotrophies there, or the nutrients that will be available in such situations when I mean, it seems that this strategy is like valid in certain steps of the infection,
right,
and depending on the pathogen, right? So did you try, or did you, your perspectives to do like intracellular pathogens also?
Right, yeah. So the biotin story is actually quite interesting because it's been kind of long valid. It's been a valid target for TB for years now. So there's been some really great work on this to show that there's a shortage of biotin as an oxytrophic in
the host.
In the host. And so, several groups have been thinking about biotin biosynthesis now, I'd say for almost 10 years as a valid target in TB. And we've actually had we had some investigation of our compound in TB, and it actually looks quite promising for TB. We've not looked in a wide variety of organisms. I mean, incidentally, we've done some work with Brian Coombs. It was published a couple of years ago, right? Looking at doing a screen, first of all, a genetic screen, and then a chemical screen for intracellular salmonella. And of course, Brian Coombs he's an expert in intracellular salmonella, and it was a really fun collaboration to work with Brian to do that investigation. I mean, one of the most interesting things to come out of that was, for me at least, is kind of a guy who doesn't think about the life cycle of salmonella very carefully very often, was that many antibiotics that wouldn't otherwise be active on gram-negative bacteria seem to work well in the intracellular situation, which suggests to me that the cells are working on the outer membrane of salmonella to allow chemicals that would otherwise be ineffective into the bug. So that was a pretty exciting takeaway there. We haven't done, but other than really those kind of two aspects, the activity of our biotin inhibitor against TB, and that work that we've done together with Brian, it's intracellularity is not something we've looked really carefully at. I do follow closely the work, for example, of the Genentech group in Staph aureus, and they have this really interesting antibody complex, rifampicin, they call it Rifalog. And if you look really closely at those papers, you see that it targets exclusively intracellular staph, and they're able to get complete sterilization with that therapy, which they're advancing along. I think it's in phase 2 right now. So it's really kind of interesting to think about, and one doesn't think about a bug like Staph aureus as being, a professional intracellular pathogen, but more and more, I think these things at least give me pause. Yeah.
And also, what about the effect of these compounds on the microbiota? Do you think that it should alter the microbiota, so it will alter also the progress of the infection?
Yeah, I mean, we think about this stuff a little bit also, and what might be the best targets in the context of not perturbing the microbiota. Yeah, and it could be that biotin synthesis is in that category. Of course, the way to answer that question is with a really good mouse model of infection. We did some work a few years ago before we sort of knew about the levels of biotin that are in the mouse, and we kind of came to the conclusion that depending on where you were in the gut, biotin might be more or less available. And some of that work, again, it was premature really to our Streptavidin human mimic experiments, but some of that work did suggest to us that there may be, that might sort of work in our favor in terms of things like vitamin and other nutrients.
Oh, that's pretty exciting, yes. And have you ever worked with mixed populations in these strategies?
You mean more than one organism, sort of microbial community type work? No, we haven't. it's, I mean, these are really interesting questions. And I think, microbiologists cleverer than I am, I think, are working on some of these problems. We're really just trying to do the straightforward kind of drug discovery stuff. We can't be good at all of it, but I'd be delighted to collaborate.
Yes, they are very complicated models anyway, but that's.
We have worked a little bit with, there's a collection of bacteria which are thought to be really important in dental caries, for example. And we've had a little collaboration around that I dare say is proven really difficult. But other than that, we haven't done very much.
Oh, that's very interesting.
Just a silly question.
Yes. Yeah.
Yes. Is this, I mean, how specific is for bacteria? Because biotin is also a vitamin for us. So how you look for specificity or for toxicity in the other case, no, with the compound, the 13772?
Right, humans don't synthesize biotin. We get it from the environment. So
okay.
Now, of course, we've done enough drug discovery in my lab to know that, you have mechanism-based toxicity where, humans might share the target, with the bacterium. And then you have all the other kinds of toxicities, which can still be problematic. But at least in this instance, we don't have mechanism-based toxicity.
Okay.
Susana, I guess you have one more question.
I'm here. Sorry. On the library you check, do you identify another target besides biotin?
So in terms of our efforts to screen for chemicals or just in general?
Yes, the chemical in the library you check.
Yeah, so believe it or not, we're still working through that list. And there's 71 compounds in there that, that are interesting. I think I showed you that 15 of them are, paramino sulfonamides. So we kind of take those out because they're not particularly novel. But, at least quite a number of compounds, more than 50, right? That paper reports on 3 compounds. It reports on the biotin compound. It reports on the PABA biosynthetic compound. And also a molecule that interrupts glycine synthesis. And a bunch of others. We're still working through all of these, believe it or not, as much as 7 years later. The glycine molecule continues to be a puzzle for us. It's a really interesting compound with great gram-negative activity that we're still puzzling about, Susanna, in terms of what the target is. So these experiments just keep going on. One of the things that my lab does a lot of is tries to come up with new platforms and approaches around generating hypotheses, often using systems techniques for what the target is of a compound. And whenever we develop a new platform, we bring all these other compounds in to say, can we make any more progress on these mechanistic studies of antibacterial action are, I think, some of the most challenging things that we can do.
Yes, certainly. Thank you. Very great, your talk.
Thanks.
Have you ever tried, I wonder, with another natural media with the uropathogens with urine, for instance?
Yeah, we've looked at this a little bit. And we've even had folks in the lab going to the restroom to create media. And I mean, there are a bunch of kind of interesting media you can think about. You can think about things, of course, for UTI. People like Marvin Whiteley have done, I think, really interesting work with sputum as a kind of you can get a synthetic sputum and you can sort of make one. I think these are all really interesting. They're very practical and very tractable to do this in vitro. But I think it's so interesting to try to get a sense of really what does the gene list look like if we ask that question in vitro. And then, of course, for us, that becomes a target list if we run that screen now with a collection of small molecules. So yeah, that's pretty much what we're doing a lot of these days.
Oh, very nice. Well, okay, we all thank you for the talk. It was great. I guess there's some other. Oh yes, Diego.
Oh no, it's
One
fine.
more
If there's no time, it's fine. I mean,
No,
it's
no,
just
it's
no.
here.
I perhaps I get
didn't see you.
you mentioned something about persisters when I asked my question, and I would like if you can add a comment on that. If you how you try with in this assay or with persister. I mean, it's so much talk about the metabolism, and so this
Right,
this dormancy and.
Yeah,
So,
it's such an interesting problem. I mean, in my view, the biggest obstacle, right, to making a lot of progress on the dormancy versus the dormancy question, I think, is it's difficult to do those experiments. Calculating CFUs per mil, right, it's a kind of a big ugly experiment that you have to do, right, if you're interested in persisters. And I think something that's kind of stands in the way of making a lot of progress in that field using, systems approaches and high throughput approaches, is coming up with different ways of asking that question that are not just CFU per mil experiments.
Excuse me, but for example, so typically when you treat with antibiotic, this definition of persistence is that you have this biphasic or two phases where you have rapid killing, and then do you see the same when you, for example, treat with this your compound?
Yeah, we do. So the biotin compound, we haven't done that experiment with our biotin compound, in fact, but again, because it's not a cytal compound, I might not.
You don't see it.
It certainly wouldn't be the first experiment we would do, but it's these are challenging experiments, though, in terms of just the time that it takes. And and talking a postdoc or a graduate student into doing these kinds of experiments is sometimes tricky.
Yeah, I can imagine. Well, thank you very much. Yeah.
Thank you.
Okay, now it's the time to get the capture, to have a souvenir of this moment. So thank you very much, Eric, once again for your very interesting talk and your work, for sharing with us all your work. So let's give
thanks so much for having us.
Kind of a couple of shots to remember this moment.
1. Two.
Are we ready?
Okay.
Okay. Thank you very much.
Thank you.
Okay. Thank you, everybody, for being here, for attending the talk. I see many well-known faces. So thank you.
Take care. Stay well, everyone.
Okay, thank you, Eric. That was very nice meeting you. And I hope maybe you will receive some mails or for contacts or collaborations. I'm sure that many of our scientists will be interested, our microbiologists will be interested in your work. So
wonderful.
That was great having you here today.
Yeah, thanks so much for having me.
Okay, thank you very much.
I wish I could have been there.
Yes. We hope that maybe next year,
it would be fun.
All these pandemics, yes.
Yeah.
It
Fingers
would be
crossed.
very, very nice to share a meeting room together.
You bet. You bet.
Okay. Thanks a lot again.
Okay. Take care.
Take care. Bye-bye. Have a good day.